All right, friends, we are back for video 2, lecture 16, and in this video we are going to talk about blood volume. So in the very beginning of our class, we covered a little bit about just fluid compartments of the body. And we talked about the 3 fluid compartments being intracellular and, or I should say, 2 big fluid compartments being intracellular and extracellular, and then the extracellular fluid compartment being further divided between the interstitial fluid, which is the fluid between the blood vessels and the cells, and the fluid inside of the blood vessels, which we know is called plasma volume. So we've got the intracellular compartment being our largest fluid compartment of the body, containing about 2/3 of our overall body water inside of our cells. And you can see on that picture there, that accounts for about 27 to 30 liters of fluid. And then the remaining fluid is outside of our cells, and that's the remaining 15 to 16 or 16.5 or so liters of fluid. The majority of that fluid is interstitial. You can see between 11 to 13 liters is interstitial, and the plasma volume is only about 3 to 3.5 liters. So of the extracellular fluid, roughly 80% of it is interstitial, and about 20% of it is found in the plasma. Now, in this video, we are going to be kind of moving towards understanding how fluid moves between these two compartments. Right, so we're going to be talking about fluid exchange between capillaries and cells. That's where we're going with this. We know that there are a bunch of forces that are going to control the movement of water between the interstitial spaces and the capillaries, and that's going to affect overall blood volume. And we know that blood volume affects venous return, because we just talked about that in our previous video. So we're going to be talking about osmotic forces that allow for the movement of water. Remember that term osmosis refers to the movement of water. So we're going to be looking at that in some detail. In this slide, we also see that fluid moves into and out of your cells as a exchange between, basically our blood. So we can say that, sort of in a nutshell, we can say that our body fluid is always circulating in the state of kind of dynamic equilibrium. We lose fluid from our blood to our kidneys. We lose fluid when we breathe out. We sweat some fluid out, and the rest leaves through our GI tract. But ideally, what comes into the body and what goes out of the body is roughly the same. And then these osmotic forces are going to be what's driving the fluid between your cells, your interstitial fluid, and back. I should say your capillaries, your interstitial fluid, your cells, and then back and forth like that. So our big conversation in this video is going to be around capillaries. So a few lectures ago, in our previous unit, we talked about blood vessels, and we know that we have arteries, veins, and capillaries. So the capillaries, of which we've got billions in our body, our capillaries are always really close to our body cells are always really close to your capillaries. So most cells are, you can see, within a millimeter distance from a capillary. And that's important because capillaries are the only vessel of our vessels in our circulatory system where we are allowed to exchange materials across the wall. And one of the reasons we are able to do that is because, one, the capillaries are very thin-walled. The capillary itself is made up of a single cellular layer, and those cells are called endothelial cells. Endothelial cells are what are put close together to one another in this tube structure. So there's an endothelial cell, and then there's a basement membrane, and that's it. So that's a very thin tube, and it's a very small and it's a very thin tube. We know that even in our continuous capillaries, there are little spaces between those individual endothelial cells, and that's what you can see here. There are these little narrow intercellular gaps. And that little gap allows for things like water and really small things to move across that capillary wall. And of course, anything that's lipid soluble is going to be able to diffuse right across the capillary wall. So these pores between our endothelial cells allow for the transport of protein-free plasma. And the reason why protein doesn't go is because proteins are too big. Proteins can't squeeze through those little pores. But things that are small and not charged, or at least small, and of course water can move right through those pores. So we want to look at the exchange of materials across capillary walls. So how materials are exchanged across capillary walls, there's a couple of different mechanisms. The most common of the mechanisms is diffusion. Diffusion, we've talked about this before. Diffusion is the passive transport of something from a high area of high concentration to low. So the ability of lipophilic, lipid-loving molecules to pass across the capillary cell wall is easy, as long as there's a concentration gradient for them. Now, lipophobic molecules, things that are not lipid soluble, still are able to pass across the walls of the capillaries because they're allowed to go through those pores, but they've got to be small, right? Not gigantic things like proteins aren't going to be able to go through, but small lipophobic molecules will be able, that we find in the plasma, will be able to travel through those pores as well. Another type of flow is something called bulk flow. I believe we talked about this previously a long time ago as well. Bulk flow is just kind of the mass movement of water and solutes across a capillary. So when we're talking about one thing specifically, we would, or a few things specifically, would be talking about diffusion. But when we talk about, the mass movement of water and whatever's dissolved in that water across the capillary that can fit through the pores, that's called bulk flow. So the terms that kind of fall under this heading of bulk flow are filtration and absorption. Filtration refers to the movement out of a capillary. So let's see if I can draw, maybe I'll draw that over here. So if this is a capillary, right? So if we move, this is the arterial end of the capillary, and that would be the venous end of the capillary. If we were to move through those pores a bunch of water and solute out, that would be called filtration. And if we were to pull in water and solute, kind of a big way, that would be absorption. So it's this, big movement. And the purpose of filtration and absorption is to be able to really distribute materials and exchange materials, between the extracellular fluids. All right, so here's a picture to just kind of get an idea of what sorts of things are we talking about moving. So you see in the little box, so you see the capillary, and you see in the little box there, we see things at the top moving across because they're lipid soluble. So these guys, oxygen, our respiratory gases, oxygen, carbon dioxide, fatty acids, steroids, and hormones, those guys are water soluble, and they're able to move right across, right? They're able to diffuse through the cell membranes. They could go out or they could come in, depending on where we are and what the concentration gradient is all about. Then we have some small things like glucose, some small water-soluble substances, glucose, sodium ions, potassium ions. Those are going right through the pore. You see, they're going right through that little pore. So that's another type of transport here. Then we have big things like proteins, those red dots in the middle of this, inside the cell. Those are big. You see them kind of being repelled right here, right? They're being repelled because they're too big to fit through there. Now there are options for getting proteins. Obviously, proteins get in and out of your blood, clearly, or we'd all be dead. So there are transport mechanisms that are able to exchange proteins across cells, but those are kind of built into the cell. That's outside of this diffusion bulk flow conversation. And then at the bottom, you can see a water-filled pore as well. Water, of course, is going to easily transport from inside and outside of those vessels freely, depending on the concentration, depending on the concentration and pressure gradients. All right, so let's talk about forces across capillary walls. So this whole conversation we could kind of call capillary dynamics. So there's basically two types of forces that are going to generate the movement of solutes or molecules across capillary walls. And those forces are one called hydrostatic pressure. Hydrostatic pressure is pressure generated by fluid. And I'm going to talk more about that in a moment. Osmotic pressure is the osmotic force that's being exerted on the water by non-penetrating solutes. So remember, water will always move towards where there is more solute. So if we have a non-penetrating solute, remember that from the beginning of the semester, a non-penetrating solute is a solute that is not able to transport from one place to another. So if you've got inside of your blood, which you do, have a lot of protein, plasma proteins, we talked about that in our 2 lectures before. Our most abundant one is albumin. As a result of the protein in your blood, that protein will exert a force on the water and it will pull water towards the protein. That force is called an osmotic force. So the big player when we talk about capillary dynamic and osmotic pressure, the big player here is the presence of proteins. Water will go towards the protein, and the proteins are, in normal circumstances, the proteins are inside the blood vessels. They're in the plasma. They're floating around in there. All right, so let's look at this now a little deeper. We're going to first look at, on this slide, hydrostatic pressure, and then we'll look at osmotic pressure on the next one. So fluid will always move from high pressure to low pressure if you have a pressure gradient. So the pressure gradient is important, right? Gradients are the things that are what are absolutely essential for any kind of transport, whether it's transport due to pressure or transport due to concentration. So in this case, we're looking at pressure because hydrostatic forces are pressure. It's pressure. It's all about pressure. And it's the pressure that's basically being generated as a result of the presence of fluid. Okay, so when, again, we look at a capillary, let me draw another one down here. So we've got our capillaries. My artistic ability is very poor to begin with, and even worse when I'm writing on something that's standing straight up. So here's our arterial end. Here's our venous end. That weird looking thing is supposed to be a capillary. All right, so we have different amounts of pressure from here to here. Now, I would like to say, where do you think the pressure is higher, on the arterial end or the venous end? Hopefully you're not having to think too hard about that, and you would hopefully be saying it must be higher on the arterial end because the pressure in general is higher in our arteries than it is in our veins. So as we go from the left side of the heart through the aorta, then through our big arteries, and then into our smaller arteries, and then into our arterioles, the pressure decreases from about 100 millimeters of mercury leaving the left ventricle to about 37 millimeters of mercury as we move from the arterial end. So let's say here 37 millimeters mercury. And then the pressure is going to continue to drop until we get to the other side here. And here we have it about 17 millimeters of mercury. So this is a gradient, 37 to 17. We have a higher pressure here than there. This pressure is generated by the fluid in this tube. Hydrostatic pressure is basically the pressure that's being exerted on the wall of the capillary by the fluid in the capillary. Now, there's also going to be pressure out here in the interstitial fluid. The interstitial fluid pressure is low. It's like basically 0 to 1 millimeters of mercury. So we have high pressure here, we have low pressure here. Why do you think the pressure is going down as you go from the arterial end to the venous end? The reason why the pressure goes down is because where you have high pressure here, it will push water out towards the lower pressure. So water gets pushed out, fluid gets pushed out, lipid soluble things get pushed out. They get pushed out, and as they get pushed out, the pressure decreases. So our hydrostatic pressures decrease as we move from the arterial end to the venous end. And they do that because water gets pushed out. So you can see the net filtration pressure is about 36 millimeters of mercury at the arterial end and about 16 millimeters of mercury at the venous end. And in that little blue box at the bottom there, I kind of explained that. Net filtration pressure is the hydrostatic pressure of the blood in the capillaries minus the hydrostatic pressure outside of the capillaries. So the pressure outside of the capillaries is at most, under normal circumstances, is at most as 1 millimeter of mercury, pardon me. So the difference on the arterial end is 37 millimeters of mercury, and on the venous end, giving these numbers, is 16 millimeters of mercury. That's your net filtration pressure. Now, remember, the hydrostatic pressure gradients are due to are a result of the force of the fluid in the tube that's being exerted on the tube. So, we're basically measuring the blood pressure of the capillaries. And the blood pressure of the capillaries, all blood pressure is it's the force that the blood is exerting on the walls of the tube. In this context, the tube is the capillary. So capillary pressure is higher when you go into the capillaries from the arterioles, and it's going to be lower when you leave the capillaries and start to go back to the heart. The pressure is going to continue to fall in the venous system until you get to, practically 0 when you get to the right side of the heart. Okay, so that's a hydrostatic pressure. Osmotic pressure is oftentimes referred to as colloid osmotic pressure. That is the osmotic force that's exerted on the water by the non-penetrating solutes, those solutes that are trapped in the vessel. Now, again, in this conversation about capillaries, osmotic pressure, or the colloid osmotic pressure, is due to the presence of proteins dissolved in the blood in the capillary. So blood plasma has higher colloid osmotic pressure than interstitial fluid. That means that there's more protein in the capillary than out. The difference between the pressure inside of the the protein inside of the capillary and the protein outside of the capillary is called oncotic pressure. So oftentimes you'll sometimes see these things used a little bit interchangeably. So oncotic pressure is roughly 25 millimeters of mercury. And again, that's the difference between the pressure generated by the protein inside the capillary versus the pressure generated by the protein outside of the capillary. So what this all means essentially is that, so I mentioned that blood contains high concentrations of proteins, while interstitial fluid contains very low concentrations of proteins. So what ultimately this means is it means that osmotic pressure is going to favor the movement of fluid into the capillaries on the venous end. So let's do our capillary again. And we're going to put all of this together in a few minutes. So here's our arterial end. Here's our venous end. Now what we're saying is there's more protein in here than there is out here. So ultimately what that means is, remember, the osmotic pressure is the osmotic force that's exerted on water by the presence of these guys. So remember, you kind of have to lay on what we just talked about. So with hydrostatic pressures, we come in here at high pressure and water's pushed out, fluid's pushed out. And the hydrostatic forces go down. When we get towards the venous end, the protein's still in here, it's stuck. So the protein is going to pull that water right back in again. That's what we're saying here. So this osmotic pressure is going to favor the movement of fluid back into the capillaries on the venous end. So you really have to kind of think about these two things together because they're both players in terms of the movement of fluid. So you could say, I'm going to say it in a minute here. So let me get you through that and just get you to that in just a second. So this type of arrangement of this type of these forces that are working on the fluid at a capillary, the forces are called Starling forces. This is not the same as Frank Starling's law of the heart. Starling's law of the heart. Starling was very involved in all of this stuff, clearly, and decided to name everything after himself. So really, you could think of this as capillary dynamics. So what we are saying here is that we have this combination of two sorts of pressures exerting push and pull at the capillary. So the Starling's forces are a combination of the hydrostatic pressure and the oncotic pressure that ultimately is going to predict how fluid is going to move across cell membranes. So there are, there's a little formula that if you've read the book, you've probably seen it. And it is basically stating that we've got, let me tell you what all these things mean here. So right here, PIF, that basically is looking at the pressure of the interstitial fluid, hydrostatic pressure of interstitial fluid on the arterial end, and this one is the hydrostatic pressure on the scratch what I was just saying. This is this big arrow and this little arrow. This is telling us that because of hydrostatic forces, we are going to be seeing more fluid pushed out on the arterial end. Then we're going to see that fluid pulled right back into the capillary because of the presence of proteins. So that little pi looking symbol there is looking at your oncotic pressure. So when you look at this, you can see that on the arterial end, we're pushing more blood out. On the venous end, we're pulling more blood in. So Starling's forces predict the movement of fluid out of the capillaries at the arterial end and into the capillaries at the venous end. So it turns out we don't actually pull all the fluid that we push out back in on the venous end of the capillaries. About 10 to 15% of it, depending on the circumstance, remains in the interstitial fluid. And that's ultimately going to get picked up by our lymphatic capillaries and take a lap through the lymphatic system and then be returned back into the venous system right at the right side of the heart. And I'll show you that picture at the end. All right, so let's look at another. So here it looks a little bit more complicated, but it's just the same thing, but now they just put the numbers in. So we see here, on the venous end, we see, on the arterial end, we see filtration pressure and absorption pressure. So we see on the venous end, the hydrostatic pressure in this picture is at 38 millimeters of mercury. The interstitial fluid pressure in that picture is 0. Inside the, as a result of filtration. Then what we're going to see, oops, what did I do? Sorry. Then what we're going to see is we're going to see absorption pressure. We're going to see on the capillary end, we see that we've got 25 millimeters of mercury due to the protein and 1 millimeter of mercury due to the pressure in the interstitial forces outside. Then when we go down to the other side, we see that the hydrostatic pressure has gone down inside the vessel, which we know, but the oncotic pressure is still the same at 25. So what's going to happen is we're going to just basically pull the fluid right back in again, right? So we've got our net filtration pressure higher on our arterial side and our absorption pressure higher on our venous side. And so what that ultimately does is it means that, and this is the important thing to remember, don't worry about all these numbers. That's not important. What's important is just that concept. So let me just kind of keep it simple for a second. Hydrostatic forces favor filtration. Osmotic pressure favors reabsorption. Our hydrostatic forces are higher on the arterial end and lower on the venous end. Our osmotic pressure stays constant, but because of the hydrostatic pressure going down, the protein, the non-penetrating solutes are going to pull the water right back in again. So this is the one that I would like you to just kind of remember. So the arterial end of our capillaries favor filtration because of hydrostatic pressure. The venous end of our capillaries favor absorption because of oncotic pressure. Or you could call that osmotic pressure. In this context, it's the same. So the presence of kind of one of the things to remember here is the presence of protein in the plasma causes the absorption. Blood pressure is what favors filtration. Blood pressure is high when you go into capillary and it dissipates as you leave the capillary. It's still not 0, but it goes down. All right, so here we have kind of the whole thing put together. So we said just a minute ago that the arterial end favors filtration, the venous end favors reabsorption. But here's the thing to remember, not all of the fluid that gets filtered gets reabsorbed. Some of it stays, and it's probably roughly about 10% of it stays in the interstitial fluid compartment. Now, if that happened all the time, your interstitial fluid compartment would continue to grow and grow and grow, and you'd swell and you'd swell and you'd swell, and you'd be like this big swollen, blob of fluid. That's not the way it works. And the reason for that is because we have a lymphatic system, which we're not going to talk a lot about. We mentioned it in the previous lecture, in the previous module, but the lymphatic system is hugely important for the returning of the fluid that leaks out of our capillaries into the interstitial spaces back to general circulation. And then at the same time plays a very important role in the immune system, because as we said previously, we've got in our lymphatic organs and in our lymph nodes, we've got a huge number of white blood cells. and their job is to kind of monitor the status of that fluid as it moves through the circulatory system. All right, so here we have, basically this setup where we've got our capillary, the net pressure, the net force is to push fluid out at the arterial end and pull fluid back in at the venous end. Not all of the fluid is pushed out, that's pushed out is brought back in. And so whatever is left is going to get picked up by the lymphatic capillaries, which are going to be kind of interwoven with the body capillaries. The lymphatic system is not a closed system, meaning that the vessels aren't all connected to one another like we see in the circulatory system of mammals like us. So the lymphatic system, the fluid that leaves the blood capillaries and moves into the interstitial spaces can get sucked into the lymphatic capillary because it's kind of this blind, vet capillary at that point. It's not hooked to something else. It's hooked on one end, but it's not on the other end. It's sort of this like dead end kind of a deal. And these lymphatics are interwoven in all of our capillary beds. And the job of the lymphatic system is to pick up that excess fluid that's filtered but not absorbed. And that turns out to be about 3 liters of fluid a day. It's a significant, think of liter bottles. That's a significant amount of fluid that is filtered but not absorbed. We filter about 20 liters of fluid a day. We absorb at the venous end of our capillaries about 17 liters of fluid a day, which gives us a surplus of about 3 liters that the lymphatic system needs to deal with and get out of the tissues, or else we continue to swell. So the job of the lymphatic system, again, is to pick up that excess filtrate, the excess filtered fluid, which we call filtrate, and ultimately return it back to general circulation. So if that doesn't happen, then we end up with swelling. And you don't need to memorize this. I just wanted to throw this out there for anyone that's interested. So if the lymphatic system cannot keep up with the fluid that's being filtered, so if we're filtering fluid in excess, or there's some problem in the lymphatic system that's impeding the flow of the fluid through that system, then you're going to get swelling. So you can see on this picture, and I believe this is a table out of the Fox book, these are just some ideas about swelling. Swelling is something that is common, and oftentimes, you have to just kind of think through it. So anytime someone has swelling, that means there's extra fluid in that zone. Now, that can be due to a lot of things. It can be one, probably most obviously due to an increase in blood pressure, pushes more fluid out, and the lymphatics can't move that fluid fast enough, and you get this kind of lag, and you get some swelling. Another thing that can happen, which if this doesn't immediately make sense to you, don't spend a lot of time worrying about it. But if for some reason now you've got protein in your interstitial fluid. Remember we said that there shouldn't be really any protein in there, very little. And if now you've got protein in the interstitial fluid, then you lose that oncotic pressure. And that makes it so we don't pull as much fluid back in. And that can happen if you have damage to capillaries and they become more permeable. That can also happen actually as a result of an increase in blood pressure because the capillary beds stretch and those pores get stretched and they get bigger. It can happen during inflammatory and injury as a consequence of injury, because the capillary walls tend to get kind of pulled apart, and then big things like proteins can leak out. Normally they don't leak out. You could have a situation where you're actually not making protein in the blood, and so your blood protein levels are low, so you lose that pressure from that respect. And then another option is you can see an obstruction in the lymphatic vessels. And there's a couple situations that, and you can also get damage to the lymphatics, or in some cases you have lymph nodes removed, like in the case of if somebody's had a mastectomy because they've had breast cancer, they'll oftentimes have to remove some of the nodes, and that impedes the flow through the lymphatic system. And so they'll oftentimes get swelling in the areas that normally would that the lymphatic system would normally move the fluid from the tissue through the nodes and ultimately back into general circulation. So you'll see, like, if somebody's had had a mastectomy and they've had a lot of their nodes removed, oftentimes their arm on the side of the mastectomy will get really swollen. They'll have to wear like pressure sleeves and things to try to keep the pressure, to try to move the fluid that way. There are some pretty nasty infectious agents. The ones that are known for this are some of the worms, the round worms. They're called nematodes. The vector is the mosquito. So this happens in most cases. We think about this as being a tropical illness because there's a lot more mosquitoes, where you have standing water and whatnot. Not to say that that's the only place that these can happen, but these nematodes, these roundworms, basically get into the body and they get into the lymphatic system and they block lymphatic drainage. And so people will get like really incredibly swollen as a result of that. So, and there's other diseases that can do that as well. Okay. So I'm actually going to stop this video. I'm not quite done with blood. I'm not quite done with this part of the regulation of blood volume, but let me see here. Oh, okay, I'm going to keep going. I just have a couple more slides. Okay, so what I want to talk about next, and I want to do this in a way that's not so overwhelming, that we're going to look at how, excuse me, the blood volume is regulated by the kidneys and by a couple hormones. And I want to say we're going to come back around and talk about these things in more detail when we do the kidney lecture. So this is just kind of a primer. So we know that the urine is obviously a way that we lose a lot of fluid. So the job of the kidneys, the kidneys do a tremendous amount of stuff for us. But one of the things the kidneys do is they regulate our blood volume. So urine, the formation of urine begins when our blood gets filtered across the glomerular wall. So we filter our fluid, our blood, across the glomerulus, which is a capillary. It's kind of an elaborate capillary, but it's still a capillary. And we filter it into the nephron. So you guys probably remember this, hopefully, from anatomy. This is like nearly impossible to draw. But so this is oops, wrong. Let's do it in red. All right, so let's do red for our blood. Okay, so we have the afferent arteriole, and we have the glomerulus, which is kind of this funny capillary ball. Then we have the efferent arteriole. All right, so blood is going to flow towards the nephron in the afferent arteriole and then into the glomerulus, and then whatever leaves the glomerulus is going to leave in the efferent arteriole. Then we have our glomerular capsule, and then we have our ugly nephron. Okay, so what I'm saying here is so whatever comes out of here, this is the end of the collecting duct, is going to go into the renal pelvis, and it's ultimately going to leave the kidneys and go into the bladder, and it's going to leave the body through the ureters, through the urethra. So it goes from the kidney into the ureter, from the kidney to the ureter, to the bladder, to the urethra, to the outside. It's urine. But the whole process starts off with filtering the blood here. And remember, filtration is a pressure-driven event where you push fluid across the walls of capillary, and the glomerulus is a capillary. So the process of urine formation begins in the kidney with filtration of the blood at the kidney capillaries called the glomeruli. Now, obviously, we're not going to, filter all of that. We filter, I should say, about 180 liters of fluid a day. None of us are urinating 180 liters of fluid a day. We urinate about 1.5 liters of fluid a day. So the difference between 180 liters being filtered and the actual excretion of urine, which is about 1.5 liters of fluid a day, means that not nearly all, actually less than 99% of the fluid we filter actually leaves in the form of urine. So most of that fluid is going to get reabsorbed, back into the blood at various locations. And we're going to talk all about that later. That's not what we're talking about right now. So the amount of fluid that's reabsorbed is controlled by a couple hormones and the sympathetic nervous system. And it's going to be controlled depending on what the body needs. So what the sympathetic nervous system is going to do is the sympathetic nervous system is going to cause a lot of things to happen. The sympathetic nervous system, we know, increases heart rate and increases the force of contraction. So it gets more blood moving through the circulatory system. We also are going to see the sympathetic nervous system cause the arterioles, these guys, that are feeding the glomerulus to dilate. So we put more blood into the glomerulus, which means we filter more. And ultimately, what that means is we're going to see an increase in the urine production with sympathetic stimulation. Now, that's a lot, and we'll talk more about that later. The kidneys are incredibly complex, but I just want you to know that generally when we think about sympathetic stimulation, we generally think about an increase in urine production. But I want to talk to you more specifically about two hormones really quickly. One is antidiuretic hormone. And the reason why I didn't give you a lot of words here is because I just want you to kind of know the basics. We're going to talk about antidiuretic hormone in a lot more detail later on. Right now, I just want you to kind of understand the relationship between the antidiuretic hormone and actually what's going on with blood volume, because that's our topic at hand. So antidiuretic hormone, you guys hopefully remember hearing about ADH. ADH is one of the two posterior pituitary hormones. It's actually manufactured in the hypothalamus, but secreted from the posterior pituitary, because remember, those two are connected. There's receptors in the hypothalamus that are sensitive to the concentration of your blood, which is the plasma osmolarity. So those receptors that are sensitive to concentrations of osmolarity are called osmoreceptors, and they're in your hypothalamus. You can see them right here. Right. So those guys are sensitive to the osmolarity of the blood. Now, the osmolarity of the blood means how concentrated it is, how much stuff is dissolved in the water. So we know that plasma osmolarity can increase or decrease depending on what you're doing. If you're intaking a lot of salt, remember, salt is our major extracellular ion, so it kind of is the biggest player in terms of osmolarity. So if you're consuming a tremendous amount of salt, you're having like salty, you're eating Pringles, you're having Doritos, you're having a lot of prepared food that has a tremendous amount of salt in it. The increase in salt would make your blood more concentrated, and that, which is what we're talking about here, it increases blood osmolarity. Your osmoreceptors say, hey, this is not right. There's more stuff in this blood than should be there. So as a result of that increase in plasma osmolarity is going to do a couple things. It's going to, one, cause the posterior pituitary to release antidiuretic hormone. What antidiuretic hormone does is it causes us to reabsorb water from the kidney tubule back into the blood. So it puts more water in there. Remember, this all started because the blood was too concentrated. So we put more water back into the blood. We dilute the blood. So the blood volume goes up because there's more water in this. This would be like if you had a glass with a little bit of salt in it and a little bit of water, right? And you tried to drink it. It's going to taste terrible. It's going to taste really salty. So what you could do is add more water to it, and that would dissolve the salt in there a little bit more. It would make it less concentrated. It still would taste salty, but not nearly as salty. But the concentration would go down, right? Because there's more fluid in there. That's the same thing that's happening there. The other thing that happens is this, when the blood is more concentrated, it triggers receptors. Those osmoreceptors make you really thirsty. And when you get really thirsty, you're drinking, I guess. Look at that. Look at the word there. They forgot to put the G on that graphic. I didn't notice it before. You're drinking. And when you're drinking, you put more water in the basically in the blood, right? Same thing. So you dilute the blood a little bit. So the role of antidiuretic hormone essentially is to increase blood volume and thereby decrease blood osmolarity. The trigger for the release of antidiuretic hormone is an increase in blood osmolarity that's picked up by those osmoreceptors in your brain. Now, if for some reason the blood became too dilute, you continue to overhydrate, or we continue to reabsorb too much, all this fluid, and the blood, becomes more dilute, then that would antagonize the release of antidiuretic hormone, and we'd see less antidiuretic hormone being released in that situation. This does get a little bit more complicated, of course, because everything can. But when the blood volume increases, and I'm going to show you this on a slide in a minute, but when the blood volume increases, like in this case, if you were really thirsty and you were hydrating to try to deal with this, or for whatever reason your blood was too dilute, there was too much water being pulled back into the vasculature, the volume of blood obviously is going to go up. And that would cause more blood moving back to the heart. And your right atria, where the blood is returning to, gets stretched. And when the right atria gets stretched, and also when the aortic arch stretches, when more blood is leaving the left side of the heart, that will inhibit the release of antidiuretic hormone, which is pretty interesting. And when the blood is hyperosmolar because of salt, we have another hormone that gets released from the right atria, and that's called atrial natriuretic peptide. It's on a slide in a minute, which also causes us to lose salt, and I'll show you that. So as per usual, there's a lot of homeostatic control around blood volume and blood osmolarity, because I said this in one of the very first lectures we gave. This is a big deal, right? This is a very big player here. Okay, so the next is a hormone called aldosterone. Aldosterone is a hormone that comes from the adrenal cortex. It's an adrenal cortex hormone, as hopefully you all remember. And what aldosterone, remember the big picture here is we're talking about blood volume. So what aldosterone is going to do is aldosterone is going to be released from the adrenal cortex, ultimately in response to low blood volume or low blood pressure, but not directly in response. So before we can get to the aldosterone, we have to preface that by talking about a hormone called renin, and then another molecule called angiotensin, and then we get to the aldosterone. So this whole thing is called the renin-angiotensin-aldosterone system. All right, so look at the top of the picture. You see the stimulus for this whole event, the activation of the renin-angiotensin-aldosterone system, is usually either a decrease in blood pressure overall in the body or a decrease in blood flow to the kidneys. Either one of those things. When either one of those things occur, that's going to get picked up in a region of the kidneys called the juxtaglomerular apparatus. And without getting too crazy, it's kind of wedged between the afferent arteriole and the distal tubule. I'll show you this in more detail later. So essentially, that area of the kidney, the juxtaglomerular apparatus, there's cells in there called JGA cells that are essentially like stretch receptors. So when there's not, and they monitor how much blood is basically in the afferent arteriole or the distal tubule. And if there's not enough blood in there because of either low blood pressure everywhere or some problem getting blood to the kidneys themselves, those cells will notice that the tube isn't being as stretched, and that will cause those cells to release into the blood something called renin. Now, renin is an enzyme, and renin gets released into the blood, and it catalyzes the reaction that converts angiotensinogen into angiotensin 1. Then we have another enzyme called the ACE enzyme, which stands for angiotensin converting enzyme. Angiotensin 1 is the ACE enzyme converts angiotensin 1 into angiotensin 2. So here we are. And then angiotensin 2 binds receptors on the adrenal cortex and causes the adrenal cortex to release aldosterone. The other thing angiotensin 2 does is it's a big time vasoconstrictor. So it will increase blood pressure everywhere in the body. But aldosterone's job is to facilitate the reabsorption of salt and water from the filtrate, from what would ultimately become urine, back into the blood. So it increases blood volume. Remember, that's the whole topic of this conversation. And when you put more fluid in the tubes, you're going to increase the pressure, because remember, blood pressure is a measure of how much force is being exerted on the wall of the vessel by the blood. And the vasoconstriction will also increase blood pressure. So this is a very potent way to increase blood pressure. And remember, the stimulus for the release of, for the activation of this RAA system is a decrease in blood pressure to begin with, right? Or decrease in blood flow to the kidneys, which would cause an increase in pressure in the kidneys. So that's a lot. That's called the renin-angiotensin-aldosterone system. And again, you're going to get to learn about it, yippee, later on in the semester as well. Okay, so truly, this is it for this lecture video. The very last hormone is the one I mentioned previously, and this hormone is called atrial natriuretic peptide, or ANP. So this hormone is all about what's going on in the at the right atria. What's happening in terms of venous return to the right atria. So this hormone gets released. It's produced by the atria. So we think about the heart as being, not an endocrine organ, but it is. We're learning more and more and more about all sorts of hormones that are produced all over the place, even in places you would not expect, like the heart. But the heart does produce this hormone, and it's called ANP. It's a peptide hormone, obviously. So what it does is it's produced by the atria of the heart when stretch is detected by either a high volume of blood returning to the heart, so an increase in venous return, essentially, So we got more blood in the wall of the heart, more blood returning to the heart, or more blood in the atria, and the walls of the atria, the right atria, stretch. That causes, so here we are, oops, there we are. So we got more blood in the atria. The atria stretch, right atria, left atria, any atria. The atria stretch, there's more blood in there, and that causes the atria to release the hormone called ANP. So what that means is we're going to ultimately need to get rid of some fluid. So how we do that is we do not, in the kidneys, we do not reabsorb sodium chloride, and water. Instead, we keep it in the kidney tubules. So we don't reabsorb it, and instead we excrete salt and water into keep it in the kidney tubules, and ultimately becomes urine. So we see an increase in urine volume. So when more fluid is leaving the body, that will ultimately mean that that's going to deplete some of the the fluid from the body, and it causes a decrease in blood volume. The other thing that will happen when the atria is stretched is the brain, that will send kind of a reflex message to the brain via the vagus. The vagus will pick that up, will send a message to the brain, and the brain will say, let's not have any more antidiuretic hormone around. We don't need any more water in here. We got plenty of water. So we see also a decrease in antidiuretic hormone, which I mentioned earlier. So ANP is essentially going to do a couple things. It's going to increase urine production in response to a stretch in the atria of the heart because of more blood going there. It's going to inhibit the secretion of antidiuretic hormone, and it's essentially acting as a physiologic antagonist to aldosterone. Kind of does the opposite of aldosterone. All right, my friends, that was almost 60 minutes of a conversation around blood volume. So I'm going to end this video now, and we'll do one last one on blood flow.